Okay, good morning, everybody. My name is Eduardo Santos. I'm a micrometeorologist at Kansas State University. I'm sorry for not being able to be in person today. I have a cold. Figured that you guys would not appreciate if I go and share my germs. I'm just working from home today. This talk, I'm just gonna have like broad talk on evapotranspiration measurements that you're performing in Kansas, and then some of the ET partitioning measurements that we're doing for this specific project. So the outline of this presentation, I'm gonna first talk a little bit about basic concepts and measurements of evapotranspiration, micrometeorological methods to study evapotranspiration, and then preliminary results that you will obtain here with our flux measurements. Okay, so evapotranspiration is a very important variable for the water cycle. You're looking at a global scale. You see that evapotranspiration is feeling this, like is the water that's evaporating from surface, and then you see that this vapor rises, form clouds, and then you have precipitation, and then you have this cycle. So some water is running off, some water is going back to the ocean through like rivers and streams, and some is recharging our groundwater and so forth. So in a smaller scale, like if you think about a watershed scale, evapotranspiration is an important variable to determine like how much water is actually left at the watershed. So you're looking at the balance there between ET and precipitation. So in a field scale, like what you're at a field scale, what you're interested here is the main thing we're interested in is that this evapotranspiration is directly related to yield, and then the balance between evapotranspiration and then precipitation or even irrigation is determine how much water is available for plants to grow, develop, and produce yield. Excuse me. If you look at the importance of ET for agriculture, let's look at how much water you have available, right, for agriculture. So this is like how much water you have in the earth. So you see like 97.4% is actually saline lakes and oceans, so not very useful. So you have about 2.6% of fresh water that's available, and then but most of this fresh water is actually frozen. So you have some of another large proportion is actually groundwater, and you have a tiny proportion that's actually readily accessible fresh water. That's like surface water in streams. Some from this readily available water have lakes and then soil moisture. Like about lakes will have a little bit more water, but quite a bit of water is actually in the soil. And then you have some water in the atmosphere, like you have water vapor in the atmosphere. And then you have rivers and biota, like that's a very, very, very small proportion of the total water. The main issue is that the agriculture uses a large amount of water, this fresh water. So you're talking about, I think 70, like sometimes 80% of this water is used for agriculture. And then you see, like you can see, like in the news that you having more and more often problems with droughts, and you have to come up with ways to better utilize this water to actually ensure the future of agriculture. All right, so what's evapotranspiration? So evapotranspiration is a term like you put together like evaporation, transpiration, and then you bind this as a single term called evapotranspiration. So you see here that you have some evaporation occurring like in a crop or on a canopy, a plant canopy, you see that some evaporation is, some water is evaporating from the soil. You have some transpiration, some water is going through these plants and then evaporating. And then some water, it's evaporating from the surface of the plant. So if you have a rain event and then those leaves are wet, eventually they're gonna dry, and then that water is moving from the liquid to the gaseous stage, due to like the energy, like sun energy, or even like the variables, wind, and then vapor pressure and things like that are controlling this rate of evaporation. Okay, so the reason why you just put everything together, like you call it evapotranspiration, you study evaporation and transpiration together, because it's hard to measure them separate. Um, um, so it's being like, what people are interested in is like how much water is being used, regardless where it's coming from. So it could be from the soil and then plants and so forth. But if you want to improve the water use efficiency of this the cropping systems, one of the logical steps is to reduce evaporation from the soil, and then we can keep the transpiration from the plants, because transpiration has it plays some important physiological roles, like like you have some cooling in the plants because you have some water removing that excess heat. You have a translocation of nutrients through the plant as the plants are pumping water from the soil to the atmosphere. But evaporation, from a production standpoint, is not doing much, not being very beneficial to you. So one logical step is to reduce this term. All right, so here's like how this graph is just showing how this transpiration and evaporation behave. So let's interpret this graph here. So we have here time scale here, we have sowing and harvest, and then here you have a partition of evapotranspiration. So as you see, like during sowing, like you don't have plants, 100% of the evapotranspiration is actually coming from soil evaporation, right? And then as the plants start to grow, you see like this is the leaf area index. So if you have more leaves, you see that the transpiration increases, and then the evaporation here, the contribution of evaporation to evapotranspiration decreases. And then as the crops senesce, you have more and more of the evaporation coming from the more of the evapotranspiration is coming from actually soil evaporation. So how do you measure evapotranspiration? So the gold standard here for these measurements is actually called a lysimeter. So lysimeter is basically a big scale. Like you have undisturbed soil that's put inside a ring, and then you see like a plant growing inside that ring, and then down here you have some usually like load cells or things that you can actually measure that weight or the mass of that ring of soil. So the main thing that's gonna cause variations in this weight here is actually water. So but then by using a lysimeter, you can determine this difference in mass are going to be visually proportional to the evapotranspiration. You see that if you can see here, that's a very complex setup. It's expensive. So we don't have a lot of lysimeters around. It's like very only very specific, very some facilities, and then also they are high maintenance instruments. Like you have usually like you have a dedicated technician to run these things. Okay, so an alternative to this lysimeters that we're using here is using micrometeorological methods. So then we're going to talk a little bit now about these micrometeorological methods. So before we do that, let's understand a little bit of the processes transporting mass and energy. So we have what's called like a molecular diffusion. Molecular diffusion, it can be easily seen like if you drop some dye in a glass of water, you see that over time that dye is going to diffuse and then spread out over that water. And that happens because you have a random motion in these molecules, and then usually what happens is that you have a flux, a net flux from the high concentration to the low concentration. And so over time, this is going to spread out. Like another example, like if you put salt in the water, like eventually that salt is going to dissolve, and then you're going to have a better distribution concentration that water. The same thing happens in the atmosphere. You can have some molecular diffusion in the atmosphere. So if you open like a flask of perfume in the classroom, you're going to eventually going to smell the perfume because that perfume is diffusing. Okay, so another process that's important for us is called like a turbulent, I guess turbulent diffusion, you can call that. And that's caused by random motions of air. So you have wind, and then you have these eddies or turbulent motions that form because different factors here. One is like, like different heating of the surface, and then you have convection going on, and then you can have mixing of the air, or other thing is like shear stress because you have high wind speeds with height, and that's going to cause shear and turbulence. You can see this like wind picking up dust, especially in Kansas, like all the time, and then you can see sometimes you can see eddies. Another thing is so interesting. My my daughter has a little bubble machine, and then we turn it on, and I was seeing the eddies around the house. It's pretty cool. So here you can see like this graph shows the relative importance of these two processes. So let's see if we can interpret this graph. So this is the height in centimeters, and you have here the relative importance of each of these processes. You see here, like when you're close to the ground, like close to the surface, you see that molecular diffusion is the predominant process. But as you go up with height, you see that molecular diffusion is not that important, but then turbulent diffusion becomes quite important. So micrometeorological methods, what they do is that they try to model this turbulent diffusion and couple this with some concentration measurements in a way that you can predict how much material, like energy or mass, is being transported from the surface to the atmosphere. Okay, so there is a network of flux towers that do these measurements around the world. So we have CO2, water vapor, and energy fluxes, and then they'll be measured continuously. And then you see here, this is called the FluxNet network. And then you see here that even of each of these triangles here represent a flux tower that are linked to this network. A few of them are actually set up over crops, not very many. Usually they try to focus on natural ecosystems. So we have I think a couple of them in the Konza, and then you see that they are not evenly distributed. You have more like in North America, Europe, Australia, and then like a few in South America and Africa. So not very evenly distributed because this is, I mean, most equipment is expensive. So here's some of the data that you actually collected over crops. So Kevin's processes this data almost every day. So here's some graphs that he plotted. So you see here, this is ET over time. And then you have the CO2 flux. So let's see if you can spot what's going on here. So we have two pictures. This picture is taking the beginning of the growing season. This one is towards the end. You see any trends there?
Trends, see anything?
Yeah.
I know what Kevin does, but you can't say. What do you know? What's the big picture kind of, a couple of big picture things that you see?
So,
It's up and down, right? And can you see how the up and down on the top relative to the up and down on the bottom? Are they the same? I look at it, so you go, got this reversed. Yeah, so they're reversed, right? So when ET is up, CO2 down and cycling.
So just to help here, negative flux it means that it's going towards the surface, and a positive flux is going away from the surface. So you
There
see,
you go.
ET has like a positive values, right? So it means that you have more water leaving the surface than going to the surface. So you see here this up and down. So this is daytime, nighttime, daytime, nighttime, and you see. But what I'm interested in showing here, like look at this trend, like what's going on here. So you see that it is getting larger and larger over time. And that's happening because the canopy is covering the ground, right? So you have more leaf area, and then you have more transpiration. And then you see here that with the CO2 is you have a more negative fluxes over time. Let's see if somebody can tell me why that is happening.
How does it go down or negative?
Yeah, why is it getting so negative over time?
More photosynthesis, like.
That's right. So we have more photosynthesis, right? You have more leaf area, more biomass accumulating, so we have more CO2 being removed from the atmosphere. Okay, so this is like you measure this over a canopy, and then you basically getting just the net flux. Net flux meaning that you have different components here. So for instance, during the daytime, this net flux is soil and plants combined. So you have some CO2 that's being released by microbes in the soil, and then even like plant respiration. And then you have photosynthesis predominant process. But during the night, excuse me, you see that the flux here is positive. So you have some of that CO2 is being released at night because you know, like there is no photosynthesis. So then the only flux that's going on here is actually we call it ecosystem respiration, which is like plant and soil respiration. So one thing that is going on, like with climate change, is that you're having warmer and warmer nights, and then you see that this flux is actually gonna it's increasing over time because you have warmer nights. So it means that some of this CO2 that the plants accumulate during the day is actually being lost during the night through respiration. Okay, but going back to evapotranspiration, so you have here like just a net flux, and what you'd be interested in is trying to partition this flux. So there is different methods to do this partitioning. So meaning like you want to determine this evaporation and then transpiration separate. We have done some work here, like I have a PhD student from Brazil that's working on this. And since we don't have a very large data set on crops yet, he worked with some data from the Konza prairie. So this is the phenocam on the side. So it's a phenological camera capturing the dynamics of vegetation. So this is by month. You see January, some snow in the ground, February, and then you start like April, seeing some green, and then August, you see some senescent over time. Okay. And then you see some snow here again in December. So if we use like a statistical approach to do this partition, we use different approach. I'm not going to get into the approach right now, but we use a statistical approach to use that to do the partitioning. And these are some of the results. We have here, so you see like this is the ratio between T and ET. So basically telling you how much of ET is coming from transpiration. And then this was a very dry year in 2017. And then you see here that sometimes you're like above 50%, like you come down when you have a drought spell, so plants cannot pump into water from the ground, and then is this up and down here. And some of the noise here is just the noise from the map. But when you look at this year here, 2019 was pretty wet. So we see that we have quite a bit of, most of the evapotranspiration is actually coming from the plant transpiration. And then you also see that some of this variation in evapotranspiration can be explained by the greenness of vegetation. So this is an index that you extract from these phenological cameras. So you see that some of that the variation can be explained by the greenness of the vegetation. So mostly here, like for a wet year, for a dry year, not so much. Like you don't have much of. I mean, the correlation is not it's not as strong. So then you also see here like a lag between this greenness. So the plants get green, and then you have an increase in T, the T contribution to the ET. And then here is like towards the end of the season, you see the senescence. So plants are getting brown, and then you don't see much of a green. So here, so then we said, well, does it work? And this is some work that we are collaborating with Dr. Partitioning. So the question is like, well, yeah, that's nice. Like there is some, does seem that the method has some correlation with this phenological camera, phenological cameras, and then other variables in the ecosystem. But does it provide good results? And then what we did is that we set up several of these microlysimeters on the site, and then we just basically, like, we would extract this. This is a PVC pipe, so we'd extract this PVC pipe, seal the bottom, and then every day you would go there and measure the weight of that or the mass of that core, and then the change in mass would be related to the evapotranspiration. So this is basically like a Boltzmann lysimeter, right? But we could deploy several of those around. And then since you don't have vegetation inside, you can assume that's this is the evaporation, and not the evapotranspiration, but mainly the evaporation from the soil. And that's how you see here, like you see, this is the microlysimeter. So there is a lot of variability in these microlysimeters, and this is the partitioning method. So you see that sometimes you have some, they come close together, but sometimes you don't get much of, I mean, that is like the microlysimeter is usually measuring higher values than this partitioning method. So we don't quite know what's going on here yet. We suspect that could be some variability in the site because you measure a very small area with the microlysimeter, and then the tower is covering a very large area. So the next steps here for this research is actually try to deploy this microlysimeter in a crop fields, and then do some additional measurements and try to figure out like what's going on here. So I think that's all I have for today, and then if you have questions, please let me know.